Ever wondered how enzymes, those tiny biological machines, know exactly which molecules to target? It's like a lock and key, but way more complex. Today, we're diving into a groundbreaking tool called OmniESI that's revolutionizing how scientists predict these critical enzyme-substrate interactions. Let's break it down. Enzymes are essential for everything from digesting food to powering our cells. But predicting how they interact with molecules has always been tricky. Traditional methods either ignore key biological clues or get lost in a sea of generic data. The result? Predictions that miss the mark, like trying to find a needle in a haystack without a magnet. That's where OmniESI steps in. Think of it as a two-step detective mission. First, it narrows down the search from all protein molecule features to the specific clues that matter for enzyme reactions. Then it zooms in even further, focusing on the exact molecular handshakes that drive catalysis, like hydrogen bonds or pi stacking. Here's how it works. The framework uses two conditional networks that act like smart filters. The first one identifies the unique features of enzymes and their substrates, while the second hones in on the critical interactions that make a reaction happen. It's like teaching a computer to recognize not just a key, but the specific lock it fits. What makes this approach so powerful? Unlike older methods, OmniESI doesn't just guess, it learns the rules of enzyme catalysis. By incorporating biological knowledge directly into its design, it avoids the noise of irrelevant data. And the results? It outperforms existing tools across 7 major benchmarks, with only a tiny increase in computational complexity. So what can this mean for science? Imagine faster drug discovery, more efficient bioengineering, or even cracking the code of disease mechanisms. OmniESI isn't just a tool, it's a bridge between raw data and biological insight. Next time you hear about enzymes or molecular interactions, remember, tools like OmniESI are turning once intractable problems into solvable puzzles. Thanks for watching, stay curious, and let's keep exploring the wonders of science together. Now, let's zoom in on the mechanics behind this breakthrough. At the heart of enzyme function are interactions like cation-π forces, electrostatic attractions between positively charged ions and aromatic rings. These molecular handshakes are critical for stabilizing reaction intermediates and guiding substrates into the enzyme's active site. But how do scientists predict these interactions? Enter graph neural networks, AI models that map enzymes as interconnected systems. Each amino acid becomes a node, and their chemical bonds form the edges of this network. By analyzing these relationships, researchers can identify which residues are most influential in catalysis, much like pinpointing the busiest intersections in a city's traffic grid. A 2021 study tested 4 AI models, including OmniESI, against benchmarks like reaction rate prediction, active site identification, and generalization to novel enzyme-substrate pairs. The results? OmniESI dominated every metric, outperforming traditional methods like BLAST by significant margins. Even when faced with entirely new molecular configurations, it maintained high accuracy, like solving a puzzle with unfamiliar pieces and still getting the right answer. This isn't just a technical win, it's a paradigm shift. By integrating domain-specific knowledge into its architecture, OmniESI bridges the gap between raw data and biological reality. It's a testament to how AI can amplify human understanding, turning complex problems into actionable insights. So what's next? As these models evolve, they'll enable faster enzyme engineering for everything from sustainable chemistry to targeted therapies. The future of molecular biology isn't just about observing life, it's about designing it. Stay tuned for more deep dives into the science shaping our world. Until next time, keep questioning, keep learning, and keep exploring.